Please welcome Professor Joel Cuello, Department of Biosystems Engineering from University of Arizona.
Thank you all very much. Good evening, salam alaikum. I guess it's too loud. All right, well, salam alaikum. Good evening to everyone. And I'd like to thank, of course, the organizers for having me here. Special thanks to Dr. Youssef Al-Hafid. So let me go back. Oh, jeez. I guess it advanced. All right. Thank you. Sorry, it started in the middle. Okay, there we go. All right. So I'm a professor of biosystems engineering at the University of Arizona in the United States. That is our campus, and of course, you're most welcome to come and visit. My distinguished industry colleagues already have talked to you about the major applications of microalgae. And so I'm going to be talking about two emerging applications of microalgae. So in other words, the technology readiness level is not that high yet. We're talking about less than 5. But they are emerging and they are potentially crucial. So I'm going to talk about two. One is microalgae for carbon capture and production of carbon-neutral cement, and the other one is using microalgae to produce molecular hydrogen gas. So let's talk about the microalgae for carbon capture and production of carbon-neutral cement. So some of you may not be familiar with this form or type or species of green microalgae. They are referred to as coccolithophores. These are microalgae actually that are green, meaning to say they're photosynthesizing. But what's unique about them is that they surround themselves with a plate that is made of calcium carbonate, which is limestone or calcite. And so that's what makes them unique. But they are photosynthesizing, so there are two processes involved. One is photosynthesis, where CO2 is fixed into carbon molecules, and then the other one is calcification. So this is the most common species in nature, Emiliania huxleyi. Here's another one, which is Coccolithus pelagicus, just to kind of show you the morphology of the plate. Again, it's limestone, it's calcium carbonate. Here's a third example, which is Chalcidiscus leptoporus. And again, you can see the individual shells or coccoliths that form the shell. So what's happening here is that, of course, this occurs in nature in the sea, in the ocean. So when CO2, and I'm sorry I can't show you the laser, but when atmospheric carbon dioxide enters the water, it goes through these reversible reactions, which are referred to as the seawater carbonate buffer system. But ultimately, a CO2 molecule is captured or is fixed by the process of photosynthesis, and it becomes part of the organic carbon within the coccolithophore. That's photosynthesis. The other process that's occurring is calcification, where you basically have two molecules of CO2. It will produce two ions of bicarbonate, and that will then produce calcium carbonate, and it will emit one CO2. So out of two molecules of CO2, one is used for calcium carbonate, the other one is re-emitted. So there is a net CO2 capture here, and that's what's outstanding about coccolithophore. So you are basically storing the fixed carbon into calcium carbonate, which is a very stable form of CO2 storage. Okay, and again, this occurs in nature, and essentially your inputs include sunlight for photosynthesis. You have seawater, which contains the calcium and other nutrients that are required by the coccolithophore, and then of course you have carbon dioxide. And that's how you form the calcium carbonate or the limestone. So this is the white cliffs of Dover, and that white material is actually limestone, and that was produced precisely through coccolithophores. And of course, that occurred in the late Cretaceous period, which is about from 100 to 66 million years ago. But this is showing you, this attests that this form of carbon storage is really stable. So if you want to fix CO2, you want to transform it into a form that will be durable and will last a long time. This is the global distribution of coccolithophores in the world's ocean. So again, sorry, I don't have my laser pointer, but if you look at Saudi Arabia on the eastern part, you have the Persian Gulf, and I guess part of that would be Oman. And you can see those blue area there, which is rich in coccolithophores. And of course, the population of coccolithophores in other parts of the ocean around the planet is quite abundant. All right, and here is in the Black Sea. This photo was taken by a satellite by NASA, and you can see those aquamarine coloration in the Black Sea, and that actually corresponds to a bloom of coccolithophores. And this occurs normally every year around June or July in the summertime. Okay, so again, this is your coccolithophore where you produce calcium carbonate using sunlight, CO2, and seawater. And so the US Department of Energy has funded a project that's being conducted at the University of Colorado at Boulder, where they are growing coccolithophores and then isolate the calcium carbonate plate so that they can isolate the limestone or the calcium carbonates. And their objective is to use that as a component for cement, because cement is 75% or thereabout of limestone, which is calcium carbonate, and about 25% clay. So today, the most common source of limestone that is used in Portland cement is coming from mining, so from quarry. But that is associated with the release of substantial amounts of carbon dioxide, including, of course, the transportation and the heating of that. So that is associated with about 2 gigatons CO2 per year in terms of cement production globally. However, if you replace the limestone that is extracted from the quarry with the limestone that is produced by the coccolithophores, then you're going to save 2 gigatons of CO2 per year. You're going to obviate the release of about 2 gigatons of CO2 per year, and that is based on the current global urban construction rate that is going on, which is equivalent to adding 1 New York City per month. So that's the rate of construction globally that is taking place. So in the United States, so this group at Boulder in Colorado, they made this calculation that to be able to meet the current US construction industry needs, they need to grow these coccolithophores in 2 million acres of ponds. So they're using open raceways or open ponds. And in terms of the land area of the United States, that corresponds to 0.5% of the total US land area, which, you know, numerically it looks fine, it looks reasonable. However, of course, in terms of applying this or implementing this, they're not talking about one single area that is 2 million acres in size. They want to break this up into smaller ponds and probably distribute them across different states where it's optimal or appropriate to grow the coccolithophores. So back to the University of Arizona, this is a colleague of mine, Professor Daniel Apayi. I'm in the Department of Biosystems Engineering. He is actually in the Department of Planetary Sciences and Astrophysics. But he's very interested in coccolithophores and the potential that it has. So he came up with this idea, and then he contacted me because I work with microalgae and I design photobioreactors. So we are collaborating now together with a few other professors at the University of Arizona. And our goal is, again, we want to mass produce the coccolithophores, but instead of using open ponds and open raceways, which will consume significant land area, we plan to grow this in photobioreactors. The growth in the photobioreactors of microalgae in general, and we expect the same thing for coccolithophores, is going to be much more efficient, so they're going to grow faster. The productivity is higher, and in the end, we're going to use a much smaller footprint of land because it's going upwards and not just area across land. So this is going to be a much more efficient method. So instead of using, say, 2 million acres of ponds to be able to meet the US construction industry needs currently, we, at least based on rough calculation, I would expect that this would be about 50% or even less of the 2 million acres of ponds if we use photobioreactors. And my laboratory group in the Department of Biosystems Engineering at the University of Arizona happens to be developing original photobioreactors that are relatively low cost but high performing, and one of these is the air accordion photobioreactor. It's made of plastic, polyethylene, and I'll talk about this in a few more minutes. And currently it's licensed by the university to a company called Algae that is based in Norway. So let's go to the second application that I want to talk about, which is microalgae for production of hydrogen. So I'm sure you're well aware that hydrogen is a renewable fuel. And there are car companies, for instance, in the mobility industry that's looking into using hydrogen fuel cells to be able to power vehicles with less emission or significantly lower emission. So there's this microalgae called Chlamydomonas reinhardti that automatically produces or spontaneously produces hydrogen gas in nature. However, the conditions need to be right for it to produce hydrogen gas. So just to a refresher here, this is your plant cell, or it could be an algae cell, and of course you have a chloroplast, which contains the green pigment called chlorophyll. So if we look at the details, you will find the chlorophyll in the membrane called granum or the thylakoids. So the thylakoids constitute the granum, and the chlorophyll is inside the thylakoid membranes. So here's another diagram of the chloroplast, and then the location of the granum, where the chlorophyll molecules are. And this is how the photosynthesis process works. So light hits the granum, the thylakoids, and that's where photosystem 1 occurs. And then there's another part of photosynthesis which is called the dark portion of the photosynthesis, where you have the fixation of CO2 into glucose or carbon molecules. So let's focus on the light-dependent reaction where the splitting of the water molecule into oxygen and hydrogen occurs. All right. So you have these two centers, which are molecules. You have PS2 on the left, that is photosystem 2. And then you have PS1 or photosystem 1 in the middle. What happens in normal photosynthesis is that sunlight at the wavelength of about 670 nanometers, it hits photosystem 2, and the electron there gets activated. The electron came from water, which split into oxygen and electrons, and that electron gets excited, and it's received by those series of molecules or electron acceptors going into photosystem 1. And then photosystem 1 receives another jolt of radiation at a wavelength of 680 nanometers. It gets excited again, and then ultimately it's received by the final electron acceptor, which is NADPH, and from there it goes into the Krebs cycle to produce glucose. Now, in normal operation, because we are producing oxygen from water, this becomes an aerobic condition. When there is oxygen, when it's aerobic, there is an enzyme called hydrogenase that is deactivated. Hydrogenase is the enzyme that catalyzes the production of hydrogen gas. So when there is oxygen in the cell, hydrogenase is deactivated or inhibited. There's no production of hydrogen gas. So, but we want to produce hydrogen gas. So how do we do this? So there's a group of investigators at the University of California in Berkeley, and the technique that they decided to employ was sulfur deprivation. So the nutrient solution where you grow the chlamydomonas, it requires sulfur because sulfur is a component of amino acids. So it's an essential part of living things. So when they deprive the culture of sulfur, photosystem 2 becomes deactivated. And so now there is no production of oxygen. And so this becomes an anaerobic condition, and hydrogenase then, which is the yellow circle to the right, it becomes activated and it catalyzes the production of hydrogen gas. All right. So the only thing with this is it's not gonna last forever. So what they do is they transfer the microalgae from the sulfur-deprived culture medium. They transfer it into a culture medium containing sulfur, all right? And then they transfer it again to one without sulfur. And that's how they're able to maintain the production of hydrogen gas. But that is a very laborious process and not very convenient. So in my case, in my laboratory, we decided to come up with a different technique. And we refer to it as the selective photosystem 2 deactivation. In this case, we're not depriving the culture medium of sulfur, but we're using light to be able to activate or deactivate photosystem 2. You see, photosystem 2 is activated at a specific wavelength with a peak at 670 nanometers. But photosystem 1 is at 680 nanometers. So what if you have a light that you only shine at 680 nanometers, it will only activate photosystem 1, not photosystem 2, because photosystem 2 at 670 nanometers. And today it's possible to do that because we have LEDs, and LEDs have very focused wavelength, peak radiation, and you can also use certain filters to really narrow down the spectrum emission of LEDs. And that's precisely what we did. And we already published the results. But anyway, what I want to tell you is that we succeeded in demonstrating this technique. That is by shining light-emitting diode with some filter at 680 nanometers, we were able to activate photosystem 1, but not photosystem 2. The end result is because photosystem 2 is not activated, there's no production of oxygen. When there's no oxygen, hydrogenase is activated and hydrogen gas is produced. So it's a great thing. And I'm not going to talk about this data, but it's just basically describing what I already told you. So the idea, of course, is to be able to grow this in photobioreactors. The most simple photobioreactor for microalgae is the bubble column, where you just have a vessel and you have a gas sparger at the bottom, and then you just bubble the gas from the bottom. The only problem is this works well when you have a small volume, but if it's scaled up, if you're talking about 1,000 liters or 10,000 liters, it doesn't work as well because the mixing, the hydrodynamic mixing at large volume is not as efficient or effective as it is in a small volume, like 1 liter. So we designed an original bioreactor we call the air accordion photobioreactor. It's also a pneumatic type of photobioreactor, meaning to say air or gas is being bubbled from the bottom. But instead of having a straight vessel, you can see the side view here, we angled it like serpentine or zigzag. And that actually improves the hydrodynamic mixing inside the photobioreactor. So you have a better mixing that is going on. And so you're able to scale up this process and maintain the growth rate and productivity of the microalgae. And that's precisely what we demonstrated with these large-scale photobioreactors. This is about 55 liters each. And again, we compared this directly with a bubble column as control. We looked at the residence time, the vessel dispersion number, the mixing time, oxygen liquid mass transfer coefficient, cell growth, and in all cases, the air accordion photobioreactor outperformed significantly the control, which is the bubble column. And that's just a demonstration going from left to right. That's a time series or a time lapse. Day one is on the left, so you have a light green. But as you go to the right, the rightmost figures is on day 10 or day 14. And you can see that it's really intense or dark green because the algae has grown significantly. All right, almost done. This is another original bioreactor that we patented. We refer to this as the accordion air loop bioreactor. And the latest one that we have is this bioreactor, which is also original. This is the 360 series internal air loop reactor. And the University of Arizona is also applying for a patent for this. And that ends my presentation. So thank you very much.